Okay, the last thing we talked about was right here, correct? We started talking about smooth muscle. And I told you this wasn't in your notes, but this is kind of like foreshadowing. Okay. Nope. Creatine phosphate is just it's like a can that holds whatever the energy is. So it just sits there, doesn't have to do anything, and then you have to have a can opener to actually open it, and that can opener is that creatine kinase, that little CK that's in. Oh, let's go back here. Where was it? I know it was here somewhere. There it is. So the creatine phosphate is kind of like this storage container. It just holds on to it. It's just a molecule with that extra phosphate on it. And then this creatine kinase is like the can opener that activates it. You don't actually need oxygen for that process. You do need water. It's a hydrolysis type of process. But with the creatine kinase, it just transfers it over. And then this empty can's just sitting around waiting to be refilled like I guess more like a Tupperware container, actually, that you can reuse over and over and over and over again. And you just keep reloading it. But this is the process that needs the oxygen, oxidative phosphorylation. So if you don't have oxygen, you can't use this one. You can use this one up here, but this goes really quick. This is like right when you step on the treadmill and you start walking, you're like, no big deal, you're not breathing heavy or anything. And then when you start breathing heavy, it's because you ran out of the creatine phosphate, and now you're depending on oxygen to give you oxidative phosphorylation with the mitochondria. You know what I mean? And then when the mitochondria starts running low, then you start getting really tired easily because you're depending on glycolysis, and you're making what nasty byproduct? Lactic acid, which makes you start feeling that soreness and that real fatigue inside the muscle. Glucose is for the last two processes. Creatine kinase doesn't need the glucose even. The glucose is for oxidative phosphorylation, where you take the oxygen plus the glucose, you send it through the mitochondria, and then you make 36 ATP. The glucose can also, if you don't have oxygen, go through glycolysis, but then the byproducts are lactic acid. So you don't need the glucose for the first one, but you do for the second two. Let's see, we started talking about smooth muscle, and I said, remember, smooth muscle and skeletal muscle are total opposites. The characteristics that make smooth muscle unique are very different, almost polar opposite of what makes skeletal muscle unique. Like skeletal muscle has striations, smooth doesn't. When we look at where you're getting calcium from, which is the physiology, where do you get calcium from in skeletal muscle? Where's the source? Sarcoplasmic reticulum. What's the main source for smooth muscle, though? The ECF, extracellular fluid, right. Do you have troponin in smooth muscle? Nope, you have troponin in skeletal, but not smooth muscle. Do you have tropomyosin in smooth muscle? Yes, but it doesn't cover the actin exactly. It's for shape, not because of it's covering the actin. And then I talked about the process here, so we already know this. Calcium comes from where? ECF. It goes in, it binds with calmodulin. Calmodulin activates this myosin kinase, which is an enzyme that phosphorylates myosin. In other words, it electrifies or energizes myosin. Now myosin is that little electromagnet with charge to it. What's it want to do? An electromagnet, once you put charge in it, it's attracted to some kind of a metal. In this situation, that myosin, now it's charged, it's magnetically drawn to actin. And it's going to stick to the actin and hold on to the actin. So the phosphorylated myosin binds with actin so the cross-bridge cycle can be completed. No two tubules. We covered this slide too, didn't we? I think we ended maybe on this one.
Yeah, that's right there.
Okay, so calcium comes in, binds to calmodulin. Calmodulin activates myosin kinase, and then myosin kinase charges the myosin, phosphorylates myosin. Okay, so smooth muscle relaxation. It's just like when we had skeletal muscle relaxation. You stop the process, everything reverts back to its original state. So if you block these little transporters out here, can calcium move in and out? Nope, calcium's blocked. Calcium wants to flow in, but when you plug up that hole, it stops flowing in. When we talked about stopping the movement of calcium in the skeletal muscle, after we stop those doors from being open, what happens next to the calcium? It gets pumped. Yep, it gets sequestered, pumped back in, right. So it gets pumped back into the sarcoplasmic reticulum. In this situation, what do you think is going to happen? We block the doorways for calcium here, now what's going to happen? Turn on a pump again, yep. So you pump it out of the cell. As you're pumping it out of the cell, it deactivates calmodulin. Calmodulin stops the whole process, and then eventually the myosin detaches from the actin. It's a slower process, though, because when you look at it, if you think of the cell, like this room, again, going back to that example, if we're talking about sarcoplasmic reticulum, every one of these tables would be like sarcoplasmic reticulum. If I dump something out, it would spread quickly across the room, right? But when we're talking about having to bring in calcium from the walls, the outer sides of the cell, the calcium leaks in here, it's going to take longer to get to the middle. It's also going to take longer to get it back out. So when you look at the process of turning off smooth, is it a fast process? No, it's slower. When we twitch our skeletal muscles, we can twitch it really quick and then we relax it really quick. When you contract a smooth muscle, it contracts slowly and it relaxes slowly. So when you think of things like your GI tract, and when it contracts, it squeezes, it doesn't go, you know, you don't poop like a rabbit, or they shoot out of you. It's a fun analogy, isn't it? Anyway, what happens is you slowly squeeze. You squeeze the food inside your GI tract, so it's kind of like mushing Play-Doh together slowly. It contracts slowly, and then it relaxes slowly. Right? Same thing with the blood vessels. They slowly squeeze, they slowly relax compared to a skeletal muscle. Okay, where are we at? Relaxation can only occur subsequent to contraction or at any time. Blah, 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 I already did this. Oh, here's the big question. So, thinking about this process, calcium channel blockers given to people with hypertension. Remember, hypertension is affecting what structures? Blood pressure, so it's affecting the blood vessels. It's what kind of muscle surrounds a blood vessel? Smooth. Not cardiac, it's smooth. So when you block the calcium channels, what's that telling you these blood vessel muscles are going to do? They're going to relax. And when they relax, the pressure makes them, I thought I heard it, dilate, or I thought I heard somebody say expand. Right. So they expand or they dilate. What's going to happen to your pressure when your blood vessels dilate? It goes down. So think about it. When you hear about a drug and they tell you what it does, like a calcium channel blocker, it blocks the calcium channels out here on the outside of the smooth muscle, preventing it from being able to do what? Constrict. It can't contract. If you don't let the calcium in, if you block the calcium from getting in, it can't contract. If it doesn't contract, that means it's in a state of what? Relaxation. Yep. If you're not contracting, you're relaxing.
The arteries of the heart, are those smooth muscles then? Are they cardiac?
The arteries coming out of the heart? Yep, the blood vessels coming out of the heart. But the heart itself is cardiac muscle.
Okay, how about like the arteries that supply the heart muscle?
The veins? Oh, are you talking about the coronary circulation? Yep, same thing. So the coronary, it's smooth too. Yep. What contracts the heart itself is cardiac, which is our next topic, but the blood vessels themselves have smooth around them. Yep. Okay, so which one of the following is true about smooth muscle contraction? How about number 4? ATP binds with actin. Has ATP ever bound with actin so far? No. So you know number 4 is wrong. How about actin binds with acetylcholine? No, not at all. Even if you were thinking about acetylcholine related to this kind of contraction, does acetylcholine go anywhere near actin? Where does acetylcholine work? It works outside the cell, right? So on the surface of the cell, actin's inside the cell. So you know number 3 has to be wrong. Number 2, myosin becomes phosphorylated. Yes, it does. Do you remember the name of the stuff that phosphorylates myosin? It's an enzyme, so what kind of word would you look for? An ACE, something ACE. So it's myosin kinase. And then, so it looks like number 2 is the right answer. How about number 1? What's wrong with it then? There's no troponin. This is skeletal muscle. So you can cross out 1, 3, and 4, and you know number 2 is the right answer. Okay, 2 types of smooth muscle. When you're looking at it, we can refer to it as multi-unit or single unit. When you're looking at multi-unit, they all work independently. So each of these little smooth muscle cells, like over here, they're all independent. This one's not touching the next one. This one's not touching the next one. They can work individually. So I can contract a little of them, or I can contract a lot of them. I don't have to contract them all at once. So when you're talking about muscles in the eye, like the ciliary muscles that pull on the lens, you can fire some of them and get a small contraction or a small pull on the lens, or you can fire a lot and get a bigger one. That's the benefit of multi-unit. You can get a strong contraction or a weak contraction. Single unit's very different. With single unit, they all work together. If you fire one, you fire all of them. It's like a chain reaction. So they fire as one single unit. How they're connected, if you remember from your anatomy, there are different ways to connect cells. You have those desmosomes that are really strong connections. You have tight junctions that are like a waterproofing, a little bit weaker, like Velcro, instead of steel and concrete. And then you have these last ones, the gap junctions. And gap junctions are little pores that connect one cell to the next. So if calcium rushes into this cell, when it gets to the other side, what's it going to do? Rush into the next cell, and then it rushes into the next cell. It'll just keep going from one cell to the next, doing what to all of those cells together? Exciting them all together. So with a single unit, you have the gap junctions that connect one to the next to the next. So when you squeeze the blood vessel, the whole blood vessel squeezes all the way around instead of just having this part that goes dimple, dimple. I want to say it makes that sound, but I have no clue. But you want the whole thing to squeeze together at one time. That's a single unit. Multi-unit, you can see places like the eye where you want sometimes a mild contraction or just a couple muscle cells to fire. But single unit, they all fire at once. The next big difference between multi and single unit, multi-unit, they're each individually controlled by neurons. This one's controlled by a neuron, this one's controlled by a neuron, that one's controlled by a neuron. So they're separately controlled by neurons. With the single unit, number one is that they're self-excitable. Do they need a neuron? They don't need a neuron. They don't need any neurons to tell them what to do. They have what we call autorhythmicity. They're autorhythmic. They can control their own rhythm. Don't misunderstand and think that they never have neurons controlling them. It's that they don't need neurons controlling them. Like, for instance, when you talk about blood vessels can be controlled by the nervous system. If you're terrified, what happens to your blood pressure? It goes up because those little smooth muscles are controlled by your brain, and they're saying squeeze. And they squeeze and they raise your blood pressure. What are they trying to do? Why are they squeezing your blood vessels? What would be the good thing about raising your blood pressure when you're terrified? You increase oxygenation to muscles and brain. When you squeeze, it's going to rush or push the blood up to your brain so you get more energy, so more oxygen and more glucose, and you can think faster, get out of a bad situation. And we'll talk about that starting today, I think, and then again in next week. But the single unit, they can be controlled by neurons, but do they need it? No, they don't need it. Multi-units need it. Multi-units, they don't talk to each other. They have to be told what to do by the neurons. Each one of those muscle cells. Single units, they can run on their own. It's just like your GI tract. Can your GI tract run without being told what to do by your brain? Sure can. Yep. You keep digesting food. You don't have to think digest, digest, digest. Okay, so multi-unit all contract individually. Single unit contract as one. Multi-unit need to be controlled by the nervous system. Single unit can have autorhythmicity or auto control. Okay, these autorhythmic ones, we're going to talk about this concept of pacemaker over and over again. But if you look, what's this dotted line when you look at a graph? It's our threshold. Down here, look, we're missing a resting potential. There's no true resting potential. These autorhythmic cells, they start doing what? Or depolarizing? They depolarize, they're getting closer to the threshold, they hit threshold, then kaboom. They have basically an action potential type of reaction. They come back into repolarization, they come down here, down to this nice low level that they're comfortable at, and then they start depolarizing again. Threshold, action potential. Repolarize, start depolarizing again. They don't ever rest completely. They always have this boom, 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 this rhythm. And it's called a pacemaker rhythm or a pacemaker potential. When you see this one cell, this leader of the pack, the pacemaker himself or herself, when this one gets fired, what are the name of these little connectors? This cell talks directly to that cell by a what? A gap junction. So as soon as it fires, it lets calcium in. The calcium's going to flow through and tell the next cell, fire. That cell tells the next cell, fire. What would this be? Multi-unit or single unit? This is one single unit. They all talk or they all act at one time. Is there a neuron controlling this? Nope. This guy is the pacemaker. It's leading the pack. It's telling it how fast to go. And so if you looked inside, you see these action potentials happening over and over and over in that one. All it takes is this one to turn all of them on at one time. Okay, so grading. How do you grade the strength of these units? So single unit smooth muscle contracts when you let cytosolic calcium flow in. What's cytosolic mean? It's the solution inside the cell. The more calcium you have in the cell, what this is telling you is what's going to happen to your contractions. The more calcium you let into the cytosol, what's going to happen to your contractions? Get smaller or get bigger? They get bigger. So you let a little bit of calcium in, you get a smaller contraction in these single units. You let a lot in, you get a bigger, more powerful contraction. And this is tone. These single units rarely are they ever completely relaxed. In fact, your GI tract lengthens about 3 to 5 more feet after you're dead because for the first time in your life, it's actually relaxed completely. So it gets longer. Otherwise, you always hold some level of tone in these smooth muscles. The same thing with your blood vessels. You're always holding some tone in your blood vessels. Otherwise, what would happen to your blood pressure? It would drop, just completely plummet. Yep. Anybody know what that's called? When you lose tone in the blood vessels so that your blood pressure rapidly drops to a fatal level? Starts with an S. Oh my God, I'm so shocked. Right, so it's shock. So when you lose the tone in the smooth muscle, these single unit smooth muscles, you get a total loss of the pressure inside that thing. Just like blood vessels. Right? So you're always holding tone. Always holding some kind of tension in here. And it's all because of the calcium. If you pumped all of the calcium out, then you would actually have no tone. But there's always calcium in there. Okay. And then modifying smooth muscle activity by the autonomic nervous system. For the multi-unit, you need it. You have to. You have to have some kind of control by the sympathetic parasympathetic nervous system. For single units, do you need it? Nope, because they have, what's the leader called? The pacemaker. Yep, so they have that pacemaker ability. So the autonomic nervous system doesn't have to, or doesn't initiate it, but it can modify it. What part of your autonomic nervous system speeds up your GI tract? Sympathetic or parasympathetic? Parasympathetic speeds it up. So what's it going to do? Is it going to let more calcium into these cells or less? The parasympathetic lets more in and they get a better contraction. You start squeezing and digesting faster. What would the sympathetic do to those calcium channels? Close them. It would close them. What's it trying to do? Slow down the contractions, right. It's trying to slow it down. So you don't need it for a single unit. You don't need the nervous system involved, but it can modify it. So here's an example. Here you have a nerve coming in, and you have these little varicosities that are going across here. So if this is a sympathetic neuron, what's the name of the chemical that's coming out of this varicosity? If this is a sympathetic neuron on the actual target muscle, what's the name of the neurotransmitter that's coming out? Norepinephrine. Right, adrenaline. If this is on blood vessels, what do you think it's going to do to most of these blood vessels? Constrict them. It turns them up and makes them squeeze, which does what to your blood pressure? Increases your blood pressure, makes it rise. If this is the sympathetic nervous system and this is a piece of your small intestine, what's it going to do? It's going to block the calcium channels and decrease its activity. So if you notice, I'm bringing back that whole nervous system thing. I told you it would keep coming back. You have to know that. If this were a parasympathetic neuron that's on the GI tract, what's the name of the neurotransmitter it releases? Acetylcholine. What's it going to do to the GI tract then? Parasympathetic's going to do what to the GI tract? Speed it up. It's going to speed up the contractions. Yep. If this is, well, I'm trying to think of a really good example. I was about to say the heart, but that's what kind of muscle would that be? That would be cardiac. Yeah. So, can't think of a really good example off the top of my head. Okay, and then smooth muscle can still develop tension. It just relaxes when it's stretched. This is kind of cool about smooth muscle. And this is a, oh, did you have a question?
And regulate and stop you from urinating and stop you from wanting to eat. So why, when somebody's like extremely scared, why do they?
It's called paradoxical defecation or paradoxical urination. The way you want to think about it is that before they were scared, they probably had to potty really bad. And so they were already on the edge, and all it took is that little oomph. Because if their bladder's really full and you scare somebody and they tense up, they tense everything up. What do they just do to everything in the abdomen? They just squeezed it, and if you start letting the urine flow, it's basically like the bladder says, what the hell, let it rip, you know? Yeah. Or if there's already feces moving into the rectum and they probably should have been in the bathroom at that point anyway, let it rip, you know? I would love to say, oh, it's evolution, it's like a deterrent, you know? Somebody scares you and you poop on their shoe and then they're like, what the? And you can run away. It doesn't work that way, though. I used to try and justify how that made sense, but it just didn't work for me. It's funny, but it does make sense.
I think you should stick with the evolution one.
Yeah, it's funnier that way, but it doesn't hold up, you know, in a medical situation. It makes people laugh, but it doesn't work so well. But yeah, that's interesting. It's because it's paradoxical. It's not a normal situation. Okay, was this the one we were on? Yeah. So smooth muscle still develops tension yet inherently relaxes when stretched. Anyway, my example with this, I love this because I know you've all heard of it before, but maybe you've never heard it explained before. You ever seen that milk gallon chug challenge? Where they try and fill up their stomach with milk as fast as they can, and what always happens? They blow chunks, right? But what's interesting is that with water, you can drink a lot of water really quickly and it just passes right through, and we'll talk about that more in the digestive tract. But when you try and drink a lot of milk, there's so many components inside the milk that the stomach stretches really fast and it doesn't like that stretch. So if you stretch it slowly, like you go to Chinese buffet, you can eat like a gallon of food, right? You just go in, and I'm sure you've all tried that before. I know I do every time I get in there. But you sit down and you slowly stretch it, and the smooth muscle will slowly relax if it's slowly stretched. So it just starts expanding, like this muscle around the stomach, which is smooth muscle. But if you rapidly try and fill it way too fast and don't give it a chance to empty, then it says, screw this. If you pull it really quick, it goes slam and pulls back. Well, think about this. If your stomach is being slammed backwards, you've got, what, 12 feet of GI tract south of the stomach, and you've got about a foot to a foot and a half north of the stomach, which is the path of least resistance. Right? So the stomach's going squeeze, 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 and you can either force it down those other 12 feet or up the foot and a half. So what happens? The foot and a half wins, right? And then it's all over the place. So if you slowly stretch a smooth muscle, it will relax to accommodate that movement. Like when you're eating things, you're eating food, your stomach smooth muscle slowly relaxes. The same thing with blood pressure. You know, you put a little bit more fluid in your body, and your blood pressure doesn't shoot up because that smooth muscle will relax to kind of accommodate it. So you drink a lot of fluids, and your blood pressure doesn't spike through the roof. As it's slowly being absorbed, you start expanding those blood vessels slowly. So it's a slow adaptation. If you try and stretch it too fast, what happens? It naturally wants to contract back. And if you want to think of it this way, it's just like that reflex we did with the knee. When you smack the patella, or under the patella, and it pulls the skeletal muscle really quick, what did the skeletal muscle want to do when you quickly jerked it? Contract back, quickly contract back. It's the same idea, different mechanisms, but the same concept. A fast movement or a fast pull of that muscle makes it want to slam back really quick. Okay, so fast stretch builds tension and it wants to snap back. A slow stretch doesn't build pressure, it slowly relaxes to accommodate. So the next time that somebody talks about that milk gallon chug, you can say, actually. Right? And then this is kind of a concept. You just have to, I started talking about it already, but smooth muscle, we call it slow and economical. Slow because does it contract quickly? Not normally. It takes a lot to make it contract quickly. When you look comparatively to a skeletal muscle I can contract and relax really lightning fast. Smooth muscle doesn't do that like that. It squeezes slow and then relaxes slow. It's a slow movement. And then we call it economical because, remember, when you want to contract a skeletal muscle, to make it contract and relax in a few milliseconds, you have to put ATP in to charge the myosin, but you also have to put ATP in to do what? Rigor mortis, think about it. To relax it too. You're using like twice as much ATP just to get a contraction and a relaxation out of a skeletal muscle. When you're looking at smooth muscle, you use an ATP to charge the myosin, it slowly binds and it pulls really tight, really slow. You get that long sustained contraction and it slowly relaxes. You get more out of your ATP with smooth muscle. And that's what they call it economical. And because of the contraction, it's slow. Remember the contraction here? 3 seconds. How many seconds did it take for a skeletal muscle in the lab last week or 2 weeks ago to contract and relax? Yeah, like a quarter of a second. So milliseconds, right? Really quick. The whole thing. This takes about 3 seconds to contract a smooth muscle and then relax it. Okay, so what do you know already about single unit smooth muscles? How about number 4? The autonomic nervous system is required for it to function. Nope, that would be multi-unit. Remember, single unit versus multi-unit. So you can cross that number 4. How about number 3? Each muscle fiber works independently of each other.
Yes.
Nope, that would be multi-unit. Number 3 is describing multi-unit, so you can cross that number 3. How about number 2? It doesn't contain pacemaker cells.
True. It does. Oh, it does. You're looking at multi-unit.
So do you need pacemaker cells for a single unit?
No.
Yes, you do. So number 2 is describing what? Multi-unit again. Number 2, 3, and 4, all 3 describe multi-units. And then number 1, does it contain numerous gap junctions? Absolutely. That's what helps them all contract as one. So number 1 is your right answer. Okay, so that was pretty quick. Smooth muscle we took care of way faster than we took care of skeletal. But you have to remember, and a lot of times when you just think in your head the comparison between the two, it'll help you. And the third type of muscle, cardiac muscle, hold on because this is a long one. So it has functional characteristics with both skeletal and single unit smooth muscle. Again, with the skeletal versus smooth, where's cardiac usually fall? Right in between. You have characteristics of skeletal. Think about its appearance. How does the cardiac muscle look? It's striated like skeletal, right? It has more of that tube-like structure, even though they're shorter cells. When you look at their function, voluntary versus involuntary, what's cardiac like? Smooth or skeletal? It's like smooth because it's also involuntary. So it has characteristics of both. We already talked about striated. The mitochondria, lots and lots of mitochondria. When you looked at the colors, what was a darker red in color? Was it the skeletal muscle or was it the smooth muscle? It was the skeletal. Why would the skeletal muscle need tons of mitochondria compared to the smooth? Because remember, smooth is very economical. It uses ATP efficiently. It doesn't need all of that mitochondria. But you have to have that power. So in this situation, what's it more like? Skeletal or smooth? Like the skeletal. And then it has T-tubules and sarcoplasmic reticulum. T-tubules makes it look like skeletal. Smooth has sarcoplasmic reticulum, but it barely uses the sarcoplasmic reticulum. And then pacemaker activity makes it look like what? Smooth. What kind of smooth? Multi or single unit? Makes it look like single unit smooth. Yep, pacemaker activity, which means it's autorhythmic. Does it have to be controlled by the nervous system then?
No.
Nope. Can your heart beat without your brain?
Yes.
Sure can, as long as you're still providing oxygen to it. And then it's influenced by autonomic nervous system, which is like what? More like smooth. There it was.
Lecture quiz tonight.
Yep, so take the lecture quiz number 4. I'm missing a pen again. Can I use your pen? Or so take lecture quiz number 4 before we come back in on Thursday. What's today's date? 2/12/13. Thank you.
Nursing today was so long. My brain is like chugging up. I think I'm going to answer this in the
Beep 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 beep.